Hello, everyone, and welcome to Sudanese Infectious Disease Association monthly grand round. It's my great pleasure to introduce our esteemed speaker for today, Dr. Hamad Abdelhadi. Dr. Abdelhadi is a senior consultant in infectious diseases at the Communicable Diseases Center of Hamad Medical Corporation in Doha, Qatar. And he is an associate professor of clinical infectious diseases at Qatar University. He's also a fellow of the Royal College of Physicians in the UK. With a diploma in HIV medicine and a master, Dr. Abdelhadi brings a wealth of expertise and experience in clinical medicine and infectious diseases. He is deeply committed to the medical education and plays a vital role in the training and development of residents and fellows in his field. In addition to his clinical work, Dr. Abdelhadi is actively engaged in academic research, leading several funded local and national projects that focus on the critical issue of antimicrobial resistance. His collaborative work with both national and international academic institutes highlights his dedication to advancing our understanding and treatment of infectious diseases. Please welcome Dr. Abdelhadi, Dr. Hamad, and the floor is yours. Thank you.
Shukran Azzaddin and Khalid and all involved in this education platform. As people sort of realize over the last year, we made a tremendous progress actually in the education program and sharing of knowledge between different regions around the world. My talk today about the epidemiology and mechanism of resistance of gram-negative bacteria in Qatar. We've been doing research for the last 6 or 7 years in Qatar. Mainly, my research interest is antimicrobial resistance, as you will see. And I sort of giving you last year, I gave you the clinical part. Now we can give you sort of the research part of the aspect of our programs. The objective of my talk mainly to raise awareness of the growing problem of antimicrobial resistance, describe the epidemiology of gram-negative bacteria presenting to secondary care, and then to familiarize with the common gram-negative pathogen associated with disease burden, mainly in Qatar and globally, and then to explore the clinical and microbiological profile of invasive multi-drug resistant gram-negative bacteria. And then after you study the microbiology, you need to look at the genomics profile because there is regional variations. And to do so, I will try to go to the basics, talk about the classification of resistant and the pathophysiology, resistant mechanism. And then I will give you an update of the epidemiology of gram-negative bacteria in Qatar. I will do this through two main big studies we've done. The first one called the SMART study, 2017 and 19, 3 years surveillance study. And then the National Priority Research Fund study. This is a very big bacteremia study, invasive MDR gram-negative bacteria. We only looked at MDR gram-negative bacteria between 19 and 21 as a continuation of the first study, but from a different perspective. So as you know, antimicrobial resistance, significant morbidity and mortality, increased management cost to healthcare, and there is a socio-economic impact. And there is lots of documents written on this. There is a global action plan in antimicrobial resistance from the WHO. The World Bank had a 2017 document. The WHO in 2017 produced what we call it priority research sort of pathogens. They have 3 categories. Priority 1, which is critical. If you look at the organism, they put on the top of it was Acinetobacter baumannii, which is carbapenem resistant, as we call it CRAB. And then number 2, carbapenem resistant Pseudomonas. And number 3 is carbapenem resistant Enterobacteriaceae, and 3rd generation cephalosporin, which is ESBL by definition. So if you look at the top pathogen, Acinetobacter, Pseudomonas, and Enterobacteriaceae, we took from this document this pathogen to study them. And then we can have priority 2 and priority 3, you can go through them. Our colleague Hatim Sati, as you looked at the posting, and this is one of the value actually of being joined, as we mentioned, being part of a team. You learn from the input from other colleagues. We know that this priority critical pathogen has been updated just recently in April of this year. And there is some movement on the top of it. Instead of Acinetobacter, they came Klebsiella pneumoniae. And this is in reflection of our clinical practice. Klebsiella pneumoniae, which is carbapenem resistant, is a nightmare, as you will see in clinical practice in ICU, and I will show you. And then you have third generation cephalosporin sort of E. coli, which is ESBL by definition. And this is a big problem. It is a very common, but we have treatment for it. For the first one, we don't have efficient treatment. And Acinetobacter now is number 3. Okay, so there is some movement of the priority pathogen priorities in 2024 study, which is more practical one. In the Gulf region, we have significant problem of MDR gram-negative bacteria. I think when I came, actually, I directed my interest and research based on what is available, and that's what people sort of, if you are wise, you direct your research to the what is available in the region. So this is a document management of infection caused by the WHO critical priority pathogen, gram-negative pathogen in the Arab countries of the Middle East. This is a consensus, as you can, all the top leaders of the infectious disease unit, Jamila Salman from Bahrain, Leila Dabbal, Matthew Basiti, as you know, all his Matthew Basiti is a very big guru in gram-negative in Italy, Watha Al-Fawzan from Kuwait, Mona Al-Musallamani from Qatar here, and leader from UAE, as well as Suha Khanj. As you know, she's a very big guru in gram-negative bacteria. From Lebanon as well as UK. You see her frequently. Faryal Khamis from Oman. So this all the top leaders of the infectious disease, they mentioned there is a need to establish effective treatment recommendation for this region, given specific challenges to antimicrobial therapy, including variation in the availability of antimicrobial infrastructure and specialist expertise. So it's a very big regional problem. I will take you to the sort of, I like always pathophysiology. If you understand the pathophysiology, the application will be very easy. Gram-negative bacteria is the sort of very big problem, multiple mechanism, as Livermore from UK saying. First, you have, the entrance through the light policy. So the entry of the antibiotics through the lipopolysaccharides mainly governed through this, what we call it, the borin channel. So special channels, which is passes the aminoglycoside, the quinolone, as well as the beta-lactam. So any modification on this borin channel by mutation might affect the diffusion of the antibiotic inside the cell. And when the antibiotic get inside the cell, we got the penicillin binding proteins, and you can call them 1, 2, 3, 4, 5. And each, for example, penicillin is specific to this penicillin binding protein. So any modification to this penicillin binding protein will cause resistance by definition. And then we have the penicillinases, which is including sort of cephalosporinases and carbaminomases, which is present in the cell wall, which inactivates these antibiotics. At the level of the DNA, we have the DNA gyrase, and the most common antibiotic affected by the DNA gyrase and topoisomerase mutation are quinolones, as we know. And then we have the ribosome, ribosome 30 and 50 subunits, and the modifying enzyme of the aminoglycoside, because aminoglycoside mainly works on the ribosomes, it will cause problem. So what happens? For example, mutation in the MXA-B and the operon M in the borin channel will affect, this is a special channel which bump antibiotic to the outside. So what happens when the bacteria come into insult by antibiotic? It will overproduce this efflux bump. And this efflux bump, for example, it happened in carbapenem, meropenem, and ertapenem, and imipenem. It will bump this antibiotic to the outside. And this is one of the difficult regions, as you can see. Sometimes you don't need to have the genotypic carbapenemases, because this either borin mutation or efflux bump mutation, which is overexpression, will lead to resistance. And then we talked about the modifying enzyme already, and we talked the target modification, like in the gyrase. And the transmission of this resistance happens between bacteria either horizontally or vertically. So vertically through chromosomes between the offsprings, or it could be, this is the most common, as you know, is plasmid is mediated from E. coli to Klebsiella, from E. coli to E. coli. So these genes can go, and this has been established for the last 30 years, that plasmid carries these resistant genes between the same species or different species, or we call it mobile genetic elements. Less likely, it could be a virus like veggies infect the bacteria with resistant gene, but this is the least common way of resistance. When we when we say MDR, what do we mean by that? So it's very important to have a criteria for antimicrobial resistance, and it's very important to look at the history. If you look at all papers, people were talking about two resistant to two drugs. In the ICMIT of 2012, the scientists and the gurus of antimicrobial resistance across the Atlantic, we've just been in the ID week, like people like Robert Bonomo and others and Tama, they sit with their European counterparts, and this is famous, what we call it, the Majarakos paper in 2012. They develop a consensus. They thought to call MDR, you have to have non-susceptibility to at least one agent from three antimicrobial categories. So, for example, if it is penicillin, this is, if it is cephalosporin, if it is gentamicin or amikacin, this is the three agent. So you have to have at least one agent from three antimicrobial classes. And to call it an XDR, it has to have an antibiotic not susceptible to one antibiotic from each class. But eventually you have to have susceptibility to two antibiotics. So this is XDR. And bound drug resistance, which is the ultimate, is resistance to all antibiotic tested one. You don't need to include the novel one because obviously always there will be new antibiotics. So if your panel have all antibiotic and all resistant, then this is a bound drug resistance. And as I can see in this vein shape, as you can see, the non-MDR are usually 60 to 70% depending where you are. And then you have the MDR, and the XDR is the smaller, and the smallest one will be the MDR. And then to the right-hand side, we always have what we mean by carbapenem resistance. The CDC produced guidance. He said resistant to any of the four carbapenems. So carbapenem, tiberpenem, ertapenem, or imipenem. This is the main sort of antibiotic use. People use ertapenem disc for resistance because ertapenem is the weakest of all. So the sensitivity will be higher. As you know, if you want to set a sensitivity, you will put it to the weakest screening tool, as you do for the VDRL, for example. It is very sensitive, but less specific. So we say that carbapenem resistance is usually associated with carbapenem resistance genes. That's why we do the PCR, for example, what we call it the carba test. But people should understand the caveat for that. The resistance to carbapenem can happen independent of the common CRGs. So this is a very important. So phenotypically, you might find another organism, it is resistant to meropenem or ertapenem, but when you do the carba test, it's negative. And that will be explained by my previous slide about the borin channels and the efflux pumps. So there is other mechanism. Unfortunately, both borin and the efflux pump, you cannot detect them by a PCR. You need to do whole genome sequence, which is more cumbersome, as you are aware. So all of you came through the Ambler classification, class A, B, C, D. In preparation, I thought, when you get older, you will get interested in history, actually. So who is Richard Ambler, who died in 2014? He was a very leading British scientist. His PhD was on molecular biology, and it was between sequencing of protein at that time, 1950, believe it or not. So his project, this is his PhD project. In the future, we might find sort of Khalid Dosa actually doing very similar things. So he did his PhD with Fred Sangler, who was a professor of molecular biology in Oxford, double Nobel Prize winner. Is the one who sequenced the insulin, okay? And then he recruited by Martin Bollocks, who was a professor of molecular biology in Edinburgh University. He was interested in beta-lactamases. That was in the 1970s. So he recruited him from Cambridge, and he asked him to sequence for him the resistance, protein. And he came with this sort of classification, sort of the serine sort of one which is class A, class C, and class D. And these, penicillinases, this is enzyme which is inactivate penicillin. You know the TEM, the CHV, sulfonyl, and CTXM, or the cefotaxime M. And then you have, it goes from green. Green means narrow spectrum. Orange mean, or amber, looks as intermediate. And all the red are very, advanced penicillinases, which is carbapenemases. So the red are carbapenemases. And this sort of one in the left, for example, the Ambler, the term, again, was a patient from Greece called Timonera. And this is, they discovered this is in the 1980s. And then mid-80s, SHB. And then the CTXM, late 1980s. And this CTXM is capable of hydrolyzing third generation cephalosporin. Okay, so by definition, they are ESBL. KBC surfaced in the 1980s. Carbo, Klebsiella pneumoniae, carbapenemase. Okay, and then you have the GES, the M, the NMC, and this is all have location. Class B, which is metallo-beta-lactamases, this is all carbapenemases. They don't have narrow spectrum, all advanced spectrum. The VIM, this is a Vietnamese sort of imipenemase. And then we have the GIM, for example, this is a German imipenemase. In 2012, there is a patient came from India, went to Sweden, and they discovered the New Delhi metallo-beta-lactamases. This was the first NDM discovery, okay? Class C is very interesting and very intriguing. We call them the AMC. This is the cephalosporinases. They inactivate cephalosporin, but they are stable against carbapenem. So AMC treatment, by definition, you see it from the guidelines, is with carbapenems, okay? I myself, I'm very intrigued because you will see all, discussion about is AMC is truly a class C or a different class. We can have a philosophical discussion about this in another discussion. And then we have class D, which is, we call them the oxazolinases, okay? They can be narrow spectrum, like OXA10 or XA1, and they can be broad spectrum. And the most common of them, anything 23 and above, is a carbapenemase. So, and the most common of them is OXA23 and OXA48. So, in summary to this, if you got interested, if you got a carbapenemases, it could be KBC, it could be class B or class D. You will not have a carbapenemase is in class C. So this is the bottom line. And this picture from the Atlas surveillance, one of the biggest surveillance study in the world. I will come to it later. And they looked at the distribution of because we are interested in understanding them, of these carbapenemases. In North America, it's very interesting where a lot of you practice. The commonest carbapenemases is KBC, so it's class A, okay? NDM on the rise. I think in the discussion recently, people discuss about that. There is NDM about 10%, okay? But the two common sort of, and there is OXA48 as well. This is in North America. In Europe, they have a mixture. They have OXA, they have KBCs. For example, Italy and Greece, big problem with KBCs. And they also have NDMs. In the Middle East, including sort of the Gulf region, we have almost 50-50 between OXA48 and NDM. Very intriguing, we don't have the KBC. I will show you later. Why that happened? Is it host factors? Is it pathogen factors? It's very interesting, actually. Is it clones? This is a research question. Nobody will be able to answer it. And in India, dominated, as you know, by NDM, which is almost 60%, and OXA48, but also they have very little KBCs, or the Indian subcontinent. So there is a geographical distribution of carbapenemases across the world. In Qatar here, I traced all the study which done in antimicrobial resistance, okay, from almost 15 years ago, 2007, 2008. For example, there is a study in bloodstream infection. ESBL was like 30% in E. coli, 20% in Klebsiella. And then you have adult admitted to ICU, 17%, stool. Nahal Tai, by the way, is a Sudanese scientist working in Qatar University. She is also interested. I work closely with her in antimicrobial resistance. Look at this, children presenting to pediatric, emergency care. 30% of children, they have an ESBL in their urine. This is a big study, 700. Very worrying. Why that happens? It's very interesting. I myself, I think there is a community prescription of cephalosporin leading to that. And this is a public health worry which need addressing. And then if you look at the urine, patient admitted, almost to 25% of them, they have ESBL. So in 2017, we launched the Global Study for Monitoring of Antimicrobial Resistant Trend. This is called the SMART study, okay? And this is SMART came to us in Qatar in 2017 when we started the collaboration, but it's one of the largest and the longest sponsored antimicrobial resistant surveillance program started in 2000. So it's almost 25 years old, okay? Uh, MSD or Merck, as known in the States, the FDA asked it to have a monitoring system when they introduced ertapenem. So it started as a regulatory requirement after the approval of ertapenem, then expanded. They found it very useful. And then if you look at the bulk of the SMART study, 500,000 isolate, 200 sites, 60 countries, all geographical locations. So I will show you the geographical location. So in the States, 21 sites, Latin America, 39, Middle East, 23, and Europe, 31 sites. Here locally, we have Jordan, Kuwait, Lebanon, Morocco, Qatar, Saudi Arabia, Tunisia, and the United Arab Emirates joined a few years ago. So it's a very, and because these companies introduce new drugs, they need to know their local pathology. Before I continue, can you hear me clearly?
Yes, we can
Yes,
hear
we
you very
can.
well.
Yeah, okay. So you will see if you put a SMART in PubMed, you will see a lot of region publish their studies. But there is other surveillance platforms. A lot of you might heard about the GLASS system, which is the Global Antimicrobial Resistance and Use Surveillance System. In Qatar, here we participated that. The WHO, they follow this GLASS system to monitor antimicrobial resistance because there is interest over the last 10 years. In European, again, they have a very good website for European antimicrobial resistance surveillance network, and you call it ECDC, very similar to the American CDC, and it will monitor resistance across Europe, and they produce yearly report, very good. Africa, they have one. In UK, they have one. The counterpart to the SMART from MSD and Merck is the Atlas, Antimicrobial Testing Leadership and Surveillance. This is the one I showed you the previous global picture, and this is sponsored by Pfizer. And then you have the Sentry. Again, if you do any research in antimicrobial resistance, you see a lot of Sentry surveillance program by the GMI laboratories. And these laboratories, because they produce techniques for antimicrobial resistance, they're interested to look at the surveillance. So our objective for the study is to survey the gram-negative bacteria associated with clinically relevant infection from various clinical specimens. And then the second objective to evaluate resistance. And the last one to examine microbiological and genetic characteristics for existing and novel antimicrobials. So this was a prospective study. It started in 2017 for 3 years. And this is the protocol. This is we call the SMART protocol. They ask you every year to produce the first 100 clinically relevant specimen from the lower respiratory tract infection. It has to be clinically relevant. You have to produce 50 from UTI, 50 from intra-abdominal infection, and 50 from bloodstream infection. Okay? Why they interested, as you can see, this is an important epidemiological thing. So they were interested more of respiratory tract infection, the VABs and the HABs, okay? So because they develop antibiotic for VABs and HABs, so this is their interest is more. So there is overrepresentation, as you can see from the lower, as you will see. So every year we produce 250, 250, and at the end of the 3 years, we have 750 clinically relevant isolates from the 4 main infection. And we do the local identification and antimicrobial susceptibility test in our lab. We have a very sort of modern lab using MALDI-TOF, and we do the ASTs by BD Phoenix. But we transfer the samples to the International Health Medical Association. This is an independent, you can read about it, central laboratory in Europe. They have 2 centers, one in America and the other one in the Switzerland. Basically, this pharmaceutical company pays this laboratory to do their tip. So they are very independent, very high quality assurance. The MIC is not used by the Phoenix. They use the Pros microdilution method, which is the gold standard for testing of ASTs. So it is very robust method. When it gives you the MIC, is better than what you have. And then the interpretation were based on the CLSI cutoff 2020, because as you see, the study finished at 2019. So for us, people will think about the pharmaceutical sort of research interaction. And this is, we showed this in the paper. They gave us access to the central database, and we extracted the result, and we've done the the analysis ourselves. So, it's very valid result with no influence of the pharmaceutical company. And for them, this is a joint benefit. Uh, some of you, if you are interested in antimicrobial resistance, you can log into the smart website. You can ask for registry, and they can give you access actually either in America or in Europe or in the Middle East. And it's very interesting. You can see of your country usually, but if you ask them to see other countries, they might allow you if you give them sort of a reasonable. And you can get a lot of research. This is like a mine sort of hub for research. Unfortunately, we don't have enough people to do research for us. So when we've done the demography, 40% of our specimen from lower respiratory tract, and I showed you selection bias, intra-abdominal infection, UTI, and bloodstream infection was the least. Okay, so the intra-abdominal and UTI was here. The predominant pathogen for respiratory, and this is, you might reflect on this in your practice, Pseudomonas on top, followed by Klebsiella. Intra-abdominal infection, the commonest pathogen E. coli. UTI, E. coli, Klebsiella, Pseudomonas, and this goes with the literature. And the bloodstream, E. coli first, because we see a lot of ESBL, and then Klebsiella, and then Pseudomonas. And where this is specimen, again, you can reflect on this in your local practice. Oh, sorry, I will go backwards. So general medicine, 40%. We are I'm a general physician, so we are very busy. And then you have the medical ICU, ED 15%, general surgery 12%. So the surgeon share of infection is less, surgical ICU 10. And as you can see, the pediatrics, as you know, this is usually healthy individual. You don't seem to have patients with significant infection like intra-abdominal or respiratory unless you have selective problems. So this is the main result. The main pathogen, E. coli, Klebsiella, Pseudomonas, Stenotrophomonas, Enterobacter, and we have only 19 Acinetobacter from all sides. Okay. When you look at the resistance, okay, this is the prevalence. If you look at the resistance, staggering resistance. This is very, sobering. Almost 60% of our E. coli from all resources were ESBL. About 4% was carbapenem resistance. The Klebsiella 40% ESBL, but 13% carbapenem resistant. So as I always teach, what you can infer from this, if you encounter an E. coli in our region, is more likely to be an ESBL. And if you encounter a Klebsiella, it will more likely to be a carbapenem resistant than E. coli. So this is why, as I showed you in the first priority pathogen, that Klebsiella pneumoniae is a very, very big problem. Also interesting, if you find about carbapenem resistant in all Pseudomonas, all our Pseudomonas isolates, this is like 160, 30%. And we've done a study with Mazin, I will show you later. This is very similar figure to the one we say, the carbapenem resistant, which is crude. So people, if you want to give carbapenem to Pseudomonas, you have to put in mind you might get it wrong in 30% of patients. Very similar figure for Acinetobacter as well. When we looked at the antibiotic susceptibility, each antibiotic to the left and to the right is a susceptibility percentage. Amikacin and the pathogen in blue is Pseudomonas, in orange is Klebsiella, and in gray is E. coli. So amikacin work very good across board. It was very interesting in UK when I was used to practice, we used to use gentamicin, not amikacin. And in the Gulf here, they use amikacin more. Better, tolerability, better adverse event, and better efficacy. It's very interesting antibiotic. It will cover all E. coli, Klebsiella, and Pseudomonas. If you look at the ceftriaxone, as I already mentioned, ceftriaxone is the good marker of ESBL. If it covers sort of, E. coli, you got 56% resistance. And for the Klebsiella, it worked better, which is very interesting. So the ceftriaxone worked better for the Klebsiella than the E. coli. Cipro, for the Pseudomonas in blue, 50-50. For the Klebsiella, 60%. And it is worse for E. coli. That's why in the Middle East here, Cipro for UTI is not a very good choice. If you look at the meropenem working for E. coli, because the majority of E. coli are ESBL, I already mentioned. So 96%. So E. coli can be covered very well with meropenem. For 13% you might get it wrong with the Klebsiella, you might get it almost 30% with the Pseudomonas. For the tazocine, tazocine, again, we have the heated discussion between carbapenem and tazocine for ESBL, but tazocine is less than the carbapenem, and we know that, okay? And then you have the tazocine for the Klebsiella, 75%, and for the Pseudomonas is almost 70%. Then we come to the novel antibiotics. This is like the kids on the blocks for us in the hospital, ceftazidime avibactam and ceftazidime tazobactam. Generally, what you can infer, they work very well for gram-negative bacteria. The ceftazidime avibactam, for example, coverage 95% for all the three organisms, which is very good. And if you look at the ceftazidime tazobactam, you can see it is coverage for the Pseudomonas is better than the Klebsiella because it's a CRE, and we will come to that, is better than the E. coli. And this is actually goes with the classical teaching that ceftazidime tazobactam, ICMIT guidelines, the IDSA guidelines. If you look at the guidelines, they put ceftazidime tazobactam mainly for Pseudomonas, including MDR Pseudomonas. They don't reserve it for the CREs, and they put ceftazidime avibactam in top of it actually. So this goes with the international literature. And then if we break them, ceftazidime avibactam and ceftazidime, and I broke them here for each organism. For example, you have the resistance rate in E. coli 4%, for the Klebsiella is 10%, because Klebsiella is more problematic. For the Klebsiella is 15%, for the E. coli 10% for the ceftazidime. So generally, you can say ceftazidime avibactam is better than ceftazidime, but I put this again in research. You have to be very transparent. They tested ceftazidime from the beginning, from the first year of the study, 2017, and they introduced ceftazidime the second year. Okay, so the number for E. coli and Klebsiella and Pseudomonas was less for ceftazidime. So although ceftazidime avibactam might appear stronger, okay, the number was less. So you have to just to take this. Mazin was, our sort of, he'd done a master's degree, then a PhD, and then now he's in Philadelphia in public health. You might introduce him actually to the group at some point because he's done a very great work with me. So in his master's degree, he did in 2012 and 2013, he done a study on ESBL, okay? But what he took 106 samples and he tested them against all antibiotics, including ceftazidime avibactam and ceftazidime. At that time, the coverage was 99%. There was only one isolate which was resistant. This is in 2012, 2013. And now look at the picture in few years' time. What happened? The resistance, it came to between 14 to 15%. Very interesting. So when we looked at Pseudomonas and as well as genetic sort of genomic classification, we have 32 Klebsiella, 22 E. coli, 7 Enterobacter ploweckii, and 9 others. This is Enterobacteriaceae. When we do the genomic study, class A was the dominant, 70%, but there was also class B. There is also class C. There is also class C. So you can see B, D, C are equal representation, but because the class A is usually ESBL, as you know, and ESBL are white, so they will be in abundance, okay? And what we've seen that in ESBL, I already mentioned that E. coli more than the Klebsiella, and cefotaxime and CTXM15 was the predominant ESBL. And this is regionally and globally. CTXM is the commonest cephalosporinases for ESBL. CREs, I already mentioned that, Klebsiella pneumoniae more than E. coli, and there is predominance of NDMs and OXA48, okay? In Pseudomonas, the main infection or the carbapenem resistant or the resistant was the BDCs. There was no MBLs. So we also had 48 CREs, mainly predominant by Klebsiella, as you know. If you look at the ertapenem resistance, 90%, imipenem resistance, 87%, meropenem resistance. So what you can infer, meropenem resistant is meropenem will perform better than ertapenem, for example. And that is exactly what I told you, that the people use ertapenem disc as a screening test. When you have resistance to all the three, it's about 70%. We have 18 from OXA48, 16 in NDM, 4 in the VIM. This is MBL. So if you combine NDM and VIM, which is both are MBL, this is 20. So you can say that it's 50-50 between OXA and NDMs and MBLs. There was only one KBC, and this is again, it's very interesting sort of observation, paucity of KBCs in our region. Our colleague Fatma Abid, she's a Tunisian who's also avid researcher with us here in Qatar, and she done a study 2014 before us, looking at carbapenemases. This is all 150 isolate, all carbapenem resistance. And if you look, she found that actually NDM was more than OXA48, and there was only 3.5% of KBCs. All these isolates were imported. So this is people who have been treated either in the UK or in the States and came back, for example, intra-abdominal infection where we isolated the KBC. So we know in our region, the KBC is very rare. I've been sort of asking Fatma for many years, to look at her numbers for bloodstream infections. Her numbers were low actually when I looked at the study between 10, because from our practice, I can see that OXA48 is more than NDM. Although in her study, because she included wound and she included urine, NDM was high. And actually, I have a study which I will show in the next paper. We answered that actually in a subsequent paper. In a bloodstream infection, in our result found that OXA48 is more than the NDM in our region in bloodstream infection. One of the interesting study outcome, they look, because as you know, MSD or Merck interested on the marketing imipenem relebactam. We don't have it available in the Gulf yet. You might have it in the States. We don't have it here. They're going to relaunch it soon over the next coming years. So they looked at 49 isolate, which was Pseudomonas, which was resistant to both imipenem and imipenem relebactam. Okay, so interestingly, there are 49. 14 of them were concordantly resistant to imipenem relebactam and imipenem. So it was resistant to both. But when they use imipenem relebactam, 35 were susceptible. So what we inferred from this observation, that imipenem relebactam restored the activity in 70% of cases. So it is an option for MDR, which is carbapenem resistant Pseudomonas. And when they looked at the genes, 10 of them was AMC, and 4 of them were, class B VIM. Imipenem relebactam does not cover, class B, so it is weak for class B, and it seems that BDCs, certain BDCs, inactivate imipenem relebactam. And this is the same 48 isolates when they tested them for ceftazidime tazobactam, because you remember this company market ceftazidime tazobactam, there was 16% resistance. And when they looked at the resistant gene, they found class B VIM, and they also looked at BDCs. Okay, so the interest, we know both ceftazidime avibactam and ceftazidime tazobactam does not cover class B, so they are weak. But the interesting is the observation that maybe the mechanism of resistance of ceftazidime may be class B. To come to the previous slide, why there is difference between imipenem and meropenem, and this is an old slide from 2006. We don't have imipenem here in the Gulf region. It's very interesting. But when I looked into this, that meropenem is more likely to resist the efflux pump. Okay, so in essence, imipenem might be better for Pseudomonas than meropenem, and I showed you the 30% resistance. And in fact, actually, the company telling me in region where they use imipenem, the incidence of the MDR Pseudomonas is less. It's an interesting observation, which is need further sort of work and research. When Mazen was with us, we produced one of the largest collection in the region, as well as actually, globally. This is 525 MDR Pseudomonas taken over 3 years. And what we've shown that if you look at the degree of resistance for meropenem and tazobactam, you're talking about 90% resistance over the 3 years for meropenem. There was some decremental. We introduced antimicrobial stewardship in 2015, and we postulated from this study because we reduced the tazocine, the MDR epidemiology reduced according to that. We have another paper on that. If you look at the Cibro, it doesn't work for MDR Pseudomonas. And the most sensitive drug was colistin. But as you know, colistin has been phased out actually for clinical practice for MDR gram-negative bacteria. And in the study of Mazin, this is the importance of research in clinical practice. We showed that the activity for ceftazim, avibactam, and ceftolozane was maximum for MDR Pseudomonas. So in 2019, we introduced in the hospital ceftazim, avibactam as a result of this study. And from the beginning of this year, we introduced ceftolozane, tazobactam. And because I showed, I talked to our, therapeutic committee. If you combine both, you will have almost 75% coverage. So the science, you should have introduced them from the beginning, two of them instead of one. And this is sort of when we've sequenced 75 of them, we found again ceftazim, avibactam, and ceftolozane, tazobactam. I invited actually Mazin to attend. I'm not sure whether he's among the audience or not, but it will be sort of interesting because this is his work. And Mazin had a colleague, PhD student with him in Sweden called Faisal, again, very clever. So we looked at the sequence type, for example, this sequence type of Pseudomonas 235, notorious MDR Pseudomonas. As you can see, it has all the resistant genes, the BDCs, the OXs, as well as this 357, which is very resistant, very resistant. And what Faisal did, he tried to find a correlation between the resistant genes and the resistant to ceftazim, avibactam, and ceftolozane, tazobactam. I put them in here. And the bigger the blob, the bigger the association. So as added in the title, we find association with Pseudomonas derived cephalosporinase 35, as well as the VEP9, as well as OXA10. This is an area of research. Nobody knows exactly now what is the exact mechanism which is drive resistant to both ceftazim and avibactam. You've seen in ICMID and ID Week a lot of posters about mechanism of resistance to these antibiotics. I think from our research, we think most likely it will be Pseudomonas derived cephalosporinases as a penicillinases. Maybe there is other mechanism, for example, mutation in the borin and the efflux bumps, but this area is still not fully explored. So, in summary, these from the SMART study, we found that the main sort of bacteria are E. coli, Klebsiella, and Pseudomonas, and the main mechanism of resistance, as I discussed, as outlined. I'm presenting two papers, so I will take a break. I think because it will be too much to discuss everything at the end, so you can ask questions regarding the SMART study, and then I will go for the next paper.
Thank you, Hamad. This was very informative and good presentation. I have just a couple questions to start with. First, I think recently our CDC here in the US, they reported that the antimicrobial resistance has increased actually during COVID by about 20%, and they thought this could be because of lengths of hospitalization and the decreased, infection prevention, because everybody was focusing on COVID and we didn't have a lot of supplies to, like for example, MRSA screening and other things were stopped during COVID, and then also increased maybe empiric antibiotic use. So did you guys seen any? I know the SMART study you said from 2017 to 2019, but do you have any indication that during COVID things are also getting worse or not? And then the other question I have is, are you guys also following any increase in fungal resistance with all, using all this broad spectrum antibiotic to to combat your resistance. Any Candida auris condition, example, in cases increasing or other resistant fungi or not?
Thank you, Zedinia. To answer to your question about the COVID and antimicrobial resistance, there is lots of papers regarding that is during the COVID there is increased resistance. Because there was liberal use of antibiotics, there is no antimicrobial stewardship, there is no enough team to cover that. And in fact, in our hospital here, we have a COVID designated hospital, which was a periphery hospital away from the central hospital where we have the bulk of antimicrobial stewardship. And that was a mistake, to be honest. You only notice it after a year, okay? And then you leave the intensivist in charge of the severely COVID patient, and they run havoc. For example, we've seen for the first time increased rise of Stenotrophomonas, including bacteremia as well as resistance. We have published studies actually with Nahla, actually from Qatar University. We showed that in Qatar here there is increased resistance during the COVID. And we have a bacteremia study with the Prof. Ali Sultan. I will come to it, 2019-21, which is bridging the COVID. We want to answer this question that there is an increased resistance during the COVID. So from the studies in Qatar, definitely we've seen increased resistance after the COVID, and it's very well documented. Regarding fungal resistance, as you know, fungal usually is less than 5% of the problem. But even if you look at the numbers, Candida krusei, tropicalis, and calabrata, we have a big problem with, Candida auris, big problem. And I think Fayham might elaborate on this on a different sort of presentation. But regionally in the Middle East, especially in Saudi Arabia, if you look at the resistant fungal infection in the Middle East, because Saudi Arabia, we're talking about 30 million. Especially fungemia. So there is, to answer to your question, there is increased rate of resistance fungi.
Ahmed Ibrahim has a question in the comment. Ahmed, you can actually, if you wanna,
Hello?
questions.
Hello?
We can hear you. Yeah.
Hello.
No, that's yeah. This is Ahmed. The slide just two slides before that about the combination percentage wise, you said you had greater coverage when you combine both. I just wanted a little bit, if you can elaborate a little bit more on that, how you found that useful to your practice.
No, I didn't mean to use both ceftazim avibactam and ceftalozone. I meant to use either or. So we looked, for example, at isolate which was resistant to one or more. So if you put the sensitivity to both, you will increase your coverage. Okay. So regarding the only combination we use as everybody use now ceftazim avibactam with aztreonam, because we don't have, as you know, to cover NDM. This is one of the recommended treatment. But again, this combination, people, they don't know to use them, what doses, which route. So this area is still not very well to cover. We don't have a very good drug to cover class B sort of carbapenemases. A big problem is in the rise to us. I think for the sake of time, if people don't, and I will just, the second, people telling me the second slide, I will just whiz through it quickly. I know we are sort of short on time. I will try to do this in the next 10-15 minutes. So we have another big trial, 2019-21. This is a national study, a prospective study. We've done this collaboration with Weill Cornell Medicine. Actually, the primary PI is Professor Ali Sultan, who's again Sudanese colleague trained in New York, and Weill Cornell, and moved to Qatar here at the inception of the medical college. In this differently, we looked at all invasive gram-negative bloodstream infection, and when we classified them according to MDR gram-negative bacteria, as by the classification I showed you. So the biggest study, we looked at all the gram-negative episode of bloodstream infection, almost 3,250. From them, we have 429 MDR isolates. We removed the duplicates, monomicrobial 366 and polymicrobial in 20s. Because of logistic region, we're not able to put all the clinical and microbiological characteristics of the rest. So we studied about 250, and we've done whole genome sequence of 100 because of restrictions of budget. We saw the budget for the research was to do 100 per year. So we found that the prevalence of MDR gram-negative is about 13%. Look at, you have almost 15% probability of having MDR in the bloodstream if you have a gram-negative. E. coli was predominant because this is mainly ESBL, 60%, 20% Klebsiella. What is different, we showed that in the paper, that we have Salmonella, it's about 7%. You don't see that in other regions. And then Pseudomonas was 5%. And we have paucity of Acinetobacter bloodstream infection. I already showed that in the poster in the ID weeks because this was a subsequent study. The sources, UTI or urosepsis, almost 50%, intra-abdominal 20%, CLABSI in 10%. In almost 40% were associated with prolonged hospital stay. And we know all the risk factors for acquisition of MDR. We know them from epidemiological studies, previous healthcare contact, previous antimicrobial steroid. Almost 50% had antibiotic in the preceding 90 days. And the mortality was 25%. This is, we call them sort of outcome things. If we look at the antibiotic, the patient, if you develop MDR gram-negative, the commonest antibiotics is carbapenems because it covers ESBL as well as aminoglycoside. But if you look at the antibiotic which they had before the hospital admission or during hospital admission, you find that cephalosporin is almost 50% of them, or beta-lactam, beta-lactamazine. I myself, I made this to our colleagues here in Qatar and in the Gulf. Believe it or not, in the Gulf here, treatment of community-acquired pneumonia is based on third-generation cephalosporin and ceftriaxone. It's very widely used, and I blame it mainly for the widespread of ESBL. And that's why in our sort of hospital, we now took it from the guidelines for community-acquired pneumonia to replace it with a penicillin in line with European and American guidelines. Because I think this is historically probably one of the things, I will try to hammer this all the time, that the cephalosporin in the community and in hospital most likely is the main drive for the ESBL. And we also looked at, this is a regression model built by our statistician. We looked at variables associated with in-hospital mortality. So we got the bifurcation is the variables, age, sex, ICU stay, duration of hospital stay, comorbidity score, and the organisms. So what we found that ICU stay, as we already know, is associated with MDR as well as mortality. When we looked at the duration of stay prolonged, if you look at the comorbidities, the more comorbidity, you are more likely to have problems. But we have association with two organisms, which is interesting, Klebsiella and Pseudomonas. And this goes actually with international sort of, I already mentioned to you, Klebsiella is problematic. And remember, if you have E. coli ESBL in the bloodstream, you treat it with carbapenem. But if you have a CRE with Klebsiella, the patient most likely will die. And we looked at the antibiotics, and I will try not to go this on details, but we had a very interesting result. Again, amikacin is very good, gentamicin is also good, meropenem good for the ESBL, but it's not good for the ertapenem, is less likely to cause problems. And then we found this sort of, for the Pseudomonas MDR, tazocine is very bad, 90% resistance. And this is, again, people should reflect when you treat a patient like this. Tigacycline, good for the E. coli, but not for the Klebsiella, and interesting. And when we looked at the MLSTs, we look at the sequencing of this genotype, we try to answer the question, is there is what we call it clonal propensity, is there is some pathogen, they have clones which might be available widely. So in E. coli, we've seen clustering of ST131 and 167. And for Salmonella, we've seen clustering of ST1. But both for the Klebsiella and the Pseudomonas, we couldn't find clustering of special sort of clones. So in a summary, from 100 whole genome sequencing, E. coli and Salmonella appear to exhibit clonal propensity. Majority of MDR Salmonella belong to the notorious ST1, and then no patent for Klebsiella pneumoniae and Pseudomonas, and then absent of invasive clones for MDR. As in the ID week, we've done with Mazin another subsequent studies with Acinetobacter, which we now ready to present. This was an overlap with this bacteremia study. And ST131 is a very well-known cause of ESBL. So most of the ESBL we see in E. coli because of this clone, most likely because of this resistant and E. coli. And out of the Salmonella, there is two major sort of global clones, ST1 and ST2. ST1 mainly in Southeast Asia, India and Pakistan. And in the study which I done with Wild Graphy, by the way, Khalid Dosa know him. I think he's bad. He's now moved to the UK again. He was a very avid researcher. We've done a great work with him. We have a 150 episode of Salmonella bloodstream infection. 10% of it were ESBL producers. So we sequenced them, these 15, and we found 14 of them were ST1s. And they carries mainly CTX-AMM. So the ESBL in Salmonella mainly carried, and we couldn't find any carbapenemases in Salmonella, which is great news. As you know, there is carbapenem resistance typhoid in Pakistan and in India, big nightmare. So we don't have it in Qatar, and it was 100% susceptible to both ertapenem and meropenem, so you can use them for treating typhoid bloodstream infection. So in conclusion, I already mentioned that the majority of bloodstream infection MDR was in adults, affecting all groups, multiple comorbidities, previous encounter with healthcare. E. coli was the most common, followed by Klebsiella, followed by Pseudomonas and Salmonella, and ESBL treated with carbapenems. MDR Pseudomonas infrequent, but associated with high mortality. And this is what we see sometimes in the ICUs, isn't it? And we found positive invasive MDR Acinetobacter, and our conclusion the same like the subsequent study we've done, that maybe there is different clones. The mortality, I know there is a lot of outcome studies in our studies to 26%, which was comparable to other regions. In some regions, the mortality of MDR maybe 50% in some cases. Last but least, we have already Ahmed sort of discussed, and Zaddin, what is the epidemiology before and after the COVID? We try to answer in subsequent work. We have to analyze, we have about 350 isolates. We've done a whole genome sequence. 150 of them are CRE. No, it's not CREs, it's 150. 150 are CREs actually, because if you add Pseudomonas, it's almost 200. So, and it's very interesting. This is going to be one of the largest, regional studies, because this is only bacteremias. And it will be interesting and very intriguing to look at the antibiotic-resistant genes in relation to microbiological characteristic and antimicrobial. So we asked our statistician to look at the antibiotic-resistant genes with the sensitive resistance, and it will be interesting to come with an answer. I'm a full-time clinician, so I wish I could have some time here in the Gulf. Unfortunately, they don't give you dedicated time for research, so I have to do this thing on top of my family commitments and work, data collection. We work with very little sort of support from biostatistician mainly through universities. We have an international sort of bioinformatics, and we collaborate with him. It's a great work. As you know, sort of this work is not a individual. We have over the last years we have a team from Hamad Medical Corporation. Imad Al-Maghboul, by the way, is Sudanese microbiologist, head of microbiology. Ali Umrani is a close colleague, senior infectious disease physician. Hussam Hawa from microbiology, Manal from microbiology. Fayha from microbiology, another colleague. Jinan was the one who collected for us the clinical. She's also Sudanese from primary healthcare. From Will Cornell, Ali Sultan, he's Sudanese professor of microbiology and immunology. Sini was his post PhD doctoral degree, and so he's biostatistician. Clement is bioinformatics. Khalid Dosa knows Mazin, who's now, as I mentioned, in Philadelphia. Have done a great work with us in Pseudomonas as well as gram-negative. Mohammed El-Brer, he worked with us in statistician. And Mohammed, he is now in Kansas City, actually. He is a budding sort of researcher, but unfortunately, he might not be directed to infectious disease. Thank you all for listening, and happy to take questions.
Thank you, Hamad. I think Dr. Fathi, do you have a question? I saw you earlier unmuted.
Yeah, well, what amazing presentation, yeah, Ahmed. Very good work, actually. I mean, multi-drug resistant is difficult to understand and difficult to treat as well. Just 2 points to bring out regarding ertapenem sensitivity in relation to other carbapenem sensitivity. I mean, we are now seeing a number of cases that are meropenem sensitive but ertapenem resistant, and this is, I think, is important for OPAT because we use ertapenem more in OPAT. So I think the recommendation now is just you don't automatically shift from carbapenems to ertapenem unless you have a sensitivity to ertapenem. So that's one point. The other point is a study which is just coming now, which will be published maybe in December this year. It is a meta-analysis of tazocine in relation to carbapenems in the use of in ESBL infections. And I think they mentioned there is no difference between the two, but you know, there are a lot of, there is only one well-controlled study, and the other 8 or 9 are, I mean, retrospective. But it is coming up. I'm interested because tazocine is commonly used in the Gulf, especially in Saudi, we use it a lot. So if they are the same, that will be a good news. Having said that, the MIC should not be more than 4 for tazocine. So these are 2 points to break up. Thank you.
Thank you, Fathi. The discussion of this area is very interesting. You mentioned about meropenem and ertapenem. From clinical practice, as you know, usually if you have, for example, patient with, sepsis, our usual practice is usually to keep the patient on meropenem and only sort of step down to ertapenem if the patient is stable. And I'm glad actually the guideline, the IDSA guideline and the ICMED guideline in support of our clinical scenario actually. This is their recommendation. I came before in a previous study, in a very interesting study about ertapenem. They looked in ertapenem for ESBL bacteremia, and they found, it was a retrospective study of almost 300 sort of patients, and they found that, there is increased mortality with ertapenem as compared to meropenem, but they are comparable in patients who had, renal impairment. So, which is, I usually take this. Maybe there is a problem with the dosing with ertapenem. Maybe once daily might not be the case. This is an area for research. Maybe you need to increase the ertapenem to higher than 1 gram. So I think that one of the weaknesses of ertapenem could be a dosing issue. And regarding what we call it the contentious discussion between tazocine and meropenem for ESBL, this is what is stemming from the Moreno's trial in the 2018. Because in the Moreno trial, they looked at patient which is cephalosporin resistance, which is either E. coli and Klebsiella, randomized to meropenem or tazocine. They found increased mortality with tazocine, and that's why you see the tazocine went off the guidelines for bloodstream infection of ESBL because of that trial. This trial was heavily contested, and because they said the selection, the cephalosporin, the testing, they included the patient which is tazocine resistant. Anyhow, when they removed the confounder and that, they found, there is no statistical difference, but still the mortality in the tazocine was higher. There is a study by Dawali, I think Mikhail Bol, she's coordinated currently a randomized controlled trials to look at this too. So the European, if you look at the IDSA guidelines, they say for ESBL bloodstream infection, you have to give carbapenems. In the European, because they are coordinating the trials, you will see them, they will say, you can use tazocine for selected type of patients. I've done a teaching on this in systematic review. Okay, if you look at retrospective, there is only one randomized control trial. So in retrospectively, they said there is no difference for tazocine against meropenem for ESBL. So it's a very contentious area. It's very interesting. My practice, which I've been using for a number of years, which is supported, that if you have a patient with, again, risk assessment, patient with ESBL, bloodstream infection or serious infection, use carbapenems. If the patient is improving, then you can downgrade. And if you're using, for example, for urosepsis, using tazocine, and the patient is improving, CRP coming down, temperature coming down, then you can, provided you're treating the patient for a short period, we're talking about less than a week, then it is safe to do that. So, but it's a very interesting area about the discussion about tazocine and meropenem.
Yeah, you're quite right. Yeah, actually, the Moreno study is the only one which is well done that is included in this meta-analysis. There are 7 or 8 other studies, but it's only the Moreno which is randomized control trial. Thank you.
Thank you so much. That is really, really good. Really good presentation and great work over the years in Doha, as always. I'm glad that Fatih have brought up the tazocine question, and that has been answered very well. The 6% salmonella that you've seen in your 100 isolates that you sequenced, very interesting, isn't it? Have you got any comment? Do you think because of over-representation of hematology or people coming from abroad, returners, travelers, or a hemato-oncology patient, because we see it more in the hemato-oncology patient, the salmonella bacteremias. So that's one preferred question. But overall, your results, especially in the main part of the presentation, it echoes very well with what I have seen here. And we have got, but shukran jazila. Thank you so much. That's great.
Thank you, Yusri. Actually, the SMART study and the bloodstream infections, the epidemiology, we both published them. So it's been published last year and this year, and we are about to publish the CREs later on this year. Saudi Arabia, by the way, is the leading country in the region, actually. And if you look at the AMR, most of the study, Hussam Zawawi, I think is a very sort of an avid researcher from Saudi Arabia. His paper on almost 15 years, and his realization of this OXA and NDM was very old. So what we can say that is existent. We see predominance of OXA and NDM. From our experience, Yusri, in our immunocompromised, it's very interesting. One of my colleague, Ali Sultan, is interested, as you know, is immunocompromised. So NDM, we see it in the immunocompromised. Very interesting. We see it in the urine bloodstream infection intra-abdominal. So there must be host issues, actually. And to answer to your question about the salmonella, the research starts with observation. Mazin was based in the laboratory, lot of salmonella bloodstream infection resistance. So he kept this is isolate, and then we studied them. 150 bloodstream infection, 15 of them were ESBL. 80% travel Yusri. So this is young people traveling and coming within a week with typhoid. So we don't see it in the context of immunocompromise. So as you know, the population here in the Gulf mainly sort of from the Asian subcontinent. And when we looked at this ST1, I found it lots of reports from India and Pakistan. And actually the bulk of patients with return travelers. There is a local sort of connection, but again, it's not a lot. But remember, the number is not very big. We're talking about 15 ESBL out of 150 salmonella. So I asked, Jawahir again, who's a sort of co-worker with Wael, it will be interesting if we look at the clinical profile, because we have them actually, we can publish them later. We have the clinical profile of this 150 patients. So we can do a bacteremia study of 150, and it will be interesting to come with other sort of observations here. One of the sort of, positive things, since I came to Qatar, we established in 2018 sort of a network of Sudanese experts in infectious diseases. And I'm very glad, actually, a lot of them were represented here, like Imad, this is the founding one with them, Ali Sultan and Mazin, sort of working between the laboratory, the university, as well as me representing the clinical part. So the idea was to collaborate together to produce work, which for the benefit for all of us. And For example, we introduce young colleagues like Wael, like Jinan, to work with us in the program to benefit from research.
Thank you for the excellent presentation, and we hope one day I will see this work also extend to Sudan.
And thank you all for attending. As we mentioned, we have to keep this sort of educational meeting going.